Hi everyone, and welcome to our 7th session on this 3-day event, the last for day 2. We are so excited to have you all here for this session on managing for better animal welfare. It is my pleasure to hand you over to the chair for this session, Dr. Chris Deskovich from the University of Queensland in Australia. Take it away, Chris.
Thank you very much, Mia, and thank you for the invitation to chair this amazing session. I'm very happy to be here. We're going to start straight away with our first speaker, and I'd love to introduce Gloeta Massie from the University of Queensland. And Gloeta is going to be talking about where's the welfare in wild rescue, rehabilitation and release.
Hi, my name is Gloeta Massie. I'm a PhD candidate at the School of Biological Sciences at the University of Queensland. And today my talk is Where's the Welfare in Wildlife Rescue? I want to acknowledge the traditional owners and their custodianship in the lands on which we meet, pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and I recognize their valuable contributions to Australian and global society. I'd like to thank the Australian government and RSPC Australia for helping fund my research. Please be aware that my presentation contains images of injured wildlife and discussions of death and suicide. Although this talk is pre-recorded, I'd like to start with some interactions that we can then talk about during the live Q&A. Please select whether you agree or disagree on the spectrum on which you agree or disagree with the following 4 statements. The first statement is, I believe that people should rescue wildlife. Second statement, I believe that people should rescue wildlife only if the wildlife has been harmed by human causes. Third statement, the primary aim of wildlife rescue should be to return the animal to the wild. Final statement, the primary aim of wildlife rescue should be to improve animal welfare. As I begin, I will use the following 2 definitions when I'm discussing these topics. What is wildlife rescue, rehabilitation, release? I'm using the definition that is providing aid to injured, orphaned, displaced, or distressed wild animals in such a way that they may survive when released to their native habitats. To talk about animal welfare, I'm using the definition that is the physical and mental state of an animal in relation to the conditions in which it lives and dies. Why study wildlife rescue, rehabilitation, and release through the lens of animal welfare? Well, this is a picture of the Melbourne cricket ground. It holds over 100,000 people. In southeast Queensland, where I did my research, and in New South Wales, the state next door, over 100,000 wild animals are reported rescued each year. We know that there are significantly more than that, but that's the number that are reported. So what I want you to do is replace every single one of these people with a wild animal. And that wild animal has been picked up by a human and brought into human care. That could be just at home, or it could be taken to a vet or some other facility. Something happens to that animal during that process, and then potentially it's released back to the wild. Consider all of the animal welfare implications of removing that animal, bringing it into a human facility, and then putting it back into the wild. My first study was a systematic scoping review. I wanted to see what was actually out there on wildlife rescue, rehabilitation, release, and whether the focus was, if any, on animal welfare. So we started with over 1,700 papers. We whittled that down to 994 papers that were actually on wildlife rescue, rehabilitation, and release. Then we further whittled that down to 152 studies based on whether there was an animal welfare type word in the title, abstract, or keywords. We collected over 7,900 data points from those studies. Then our 152 study papers, there were 3 classes, 34 orders, 61 families, and 126 genera, and 184 species studied. Now, the majority of species, 108, were only seen in 1 study. Whereas these 2 species, koalas and the northern goshawks, both had 7 studies each. They were very, very loved. 21% of all study species were birds of prey. So we're seeing a bias towards specific species and specific genre. Some of the data points we collected were how robust were the studies? How many had controls? How many had randomization? How many had blinding? Only 36% of the studies reported a control. Only 13% reported randomization, and only 1% reported blinding. There was only one study out of 152 that had a control, randomization, and blinding, which are considered to be gold standard for interventional studies. We also checked to see how many papers reported basic animal demographics, so age and sex. Here we had better results. 83% of papers reported the age and 59% reported the sex. Having said that, that age could literally be one of the study animals. They didn't have to report for all study animals. They didn't have to report medians or averages or anything. If they merely said this was a juvenile animal, we said that's reported. So we had a very lenient bar. And even then with that very lenient bar, 17% did not report any type of age and 41% did not report any type of sex. Unfortunately, when we started to look at animal welfare indicators, the situation reversed. So positive, 74% of papers reported some type of behavior. But to tell you how lenient we were being, if they even said something like, we observed an animal leaning, something that simple, even if we said they reported behavior. But when we actually went and checked, of those 74% of papers, only 2 presented an ethogram. So they might make a comment about behavior, which we said then behavior was reported. But if they actually provide how they were assessing behavior, no. The other categories, not so good. So weight, 47% reported. Body condition index, 28% reported. Housing, food, water, 34%, 41%, 21%. Enrichment, 31%. And then just even the word welfare. Out of these papers, how many even just use the word welfare? Only 30%. So my next study was a survey. Had the literature, what do the people think about wildlife rescue? So I did a snowball survey across Australia. And I had over 600 people respond to the survey, although not everyone responded to every question. So 524 people answered this question. How many wild animals do you think are rescued each year across Australia? Now 37% said more than 125,000. But 62% of respondents underestimated the number of wildlife rescued. So the majority of people underestimated how many wildlife are being rescued in Australia each year. Just to give you an idea of the scale, that little pink area represents Southeast Queensland, and over 50,000 wild animals are rescued and brought into wildlife hospitals in that pink area each year alone. Imagine then the number across the entire country. And yet, majority of respondents thought there were fewer than 125,000 animals rescued across Australia. We also asked people what they think happens to the majority of wildlife. So the reality is the majority of wildlife do not survive the experience of being rescued, rehabilitated, and released. They just don't make it to the release phase. So out of 100,000 animals, between 66 to 74% will be euthanized. So if you picture that stadium, 2/3 plus will be euthanized, and the remaining animals will be released back to the wild. This is the exact opposite of what the surveys thought. So 66% of the surveys, that's basically the blue and the orange boxes, think that the majority of wildlife survive and either are released back to the wild or are kept in permanent care. The final question from the survey I'd like to address here is how people would choose to triage rescued wildlife for care in a veterinary facility, right? So if you have multiple animals arrive, how do they triage and treat them? The largest grouping, 44%, believed that the most injured animal should be treated first. 28% of people, however, prioritized endangered species first. 14% prioritized animals that were most likely to survive first. And only 9% of people focused on treating animals that needed to be euthanized first. Which begs the question, and I'm going to ask you to think about this. If an animal is in desperate need, right, to the point that it needs to be euthanized, how many people are deprioritizing that? And where would you deprioritize, or would you deprioritize that animal over an endangered animal that was less in need? The next study that I did involved embedding myself in facilities that treat rescue wildlife. I wanted on-the-ground data for real-time arrival and time to triage and time to treatment data. I did this for over 900 animals over 40 4-plus-hour observation blocks. This picture is of a lorikeet I observed chewing its way out of a box while it was waiting to be seen, and as with the majority of rescue wildlife, euthanized. Now, I need to warn you that the next image is quite graphic. So if you are not prepared for a very graphic image of an injured animal, I would suggest you turn away from your screen or just close your eyes for the next minute. When I'm talking about animals that are waiting for triage and for treatment, I wanted to show you the types of injuries that the animals are experiencing. So the previous picture was of a lorikeet that was trying to chew its way out of its box. This picture is of an ibis. This ibis has a compound fracture, an open fracture on its wing. It arrived in a box for which it was too big, and by the time it was finally euthanized, which was the outcome for this animal, it had waited several hours in transportation to the facility where I was doing the observations. So the times that I'm going to show you do not include any of the time prior to my observations beginning. So I want to emphasize that the times do not include the time of rescue, the time that that person transported the animal to a vet clinic or to a hospital or kept an animal at home. Any of that pre-arrival time is not included in the following data. But what my observations revealed were the following data. This is still preliminary data, so it could change before it's eventually published, but this is just the broad data. On average, animals are waiting 48 minutes, these numbers, this is in minutes and hours, 48 minutes to receive anesthesia, and 53 minutes to receive water or analgesia. So it was one of those two outcomes for an animal that arrived at the facility where I was doing observations. The median time of waiting from entry into the door until receiving anesthesia was 34 minutes or 38 minutes for water and analgesia. The maximum time I observed an animal waiting for anesthesia was 6 hours and 22 minutes. And to receive water or analgesia, 5 hours and 1 minute. So again, I want you to imagine the lorikeet in the box or that ibis with the open compound fracture and put it into one of these categories for wait times. And then add on all of the time that I was not able to account for prior to arriving at the facility. My last study, and what was not expected when I started my research, but I had to include it, was the perspectives of the veterinary professionals who work in wildlife and rehabilitation. And for this, I collected over 40 hours of interviews with veterinary staff and volunteers. And again, heads up, the next picture is graphic in a slightly different way, but it's about death. So when I say that there are euthanasia rates, it's important that you actually picture what that means. This is what the daily life is for somebody who is working as a vet with rescued wildlife. When I arrived at this facility, there were no animals on that towel. Within the first hour, these were the animals that the staff had to euthanize to put on the towel. One of the things I realized in interviewing the vets and the vet staff was how much their welfare was tied to the welfare of the animals for which they are caring. They were intrinsically linked. One day I watched a vet euthanize 16 animals in a row. I want you to picture that for a second, having to basically kill 16 animals in a row and what that does to you mentally. And then how do you process the next animal that comes through? And so sometimes vets may choose to not euthanize an animal because they simply can't kill another animal that day. Conversely, sometimes the facility may be so full, an animal that might have otherwise survived will have to be euthanized because there are no facilities to care for it. All of this takes a toll on the vets themselves, and it's something we need to consider when we're talking about wildlife rescue. In closing, I challenge you to think about how could we use the pillars of sustainability, social equity, economic viability, and environmental protection to improve the wildlife rescue system and the welfare of the animals and the people involved. Thank you for your time.
Thank you, Gloeta. Very eye-opening, really important work. Our next speaker is Michelle Campbell from the Animal Welfare Unit at the Department of Regional New South Wales in Australia. And Michelle's also going to be talking about wildlife welfare, and particularly she's going to be talking about snake welfare, moving beyond controlled deprivation. Thank you, Michelle.
Hello. In this presentation, my co-author Matthew Crane and I aim to explore the contemporary scientific understanding of snake welfare and what we can do to encourage snake carers to move beyond some historical practices that have been described as nothing more than controlled deprivation. Before we get into it, we'd like to acknowledge that we stand on country which always was and always will be Aboriginal land. We acknowledge the traditional custodians of the land and waters and show our respects for elders past, present and emerging. So to begin, please take a second to think about this animal. What is your instant reaction to this image? Do you think this animal is aware of its environment, capable of conscious thought, able to have pleasurable and positive experiences, capable of experiencing fear, capable of suffering? Or perhaps you would prefer to consider those questions at more of a distance.
Snakes are an ancient group of animals, and we humans have developed a complicated relationship with them. We find them intriguingly fascinating and can at the same time be truly phobic of them. Many have the capacity to harm us, and there are various theories regarding the origins of human tendencies to avoid them. Decades of anthropocentric bias, whereby we have valued species more closely aligned with human capabilities and likeness, have been used to justify historical beliefs regarding the simplicity of the reptilian brain and consequently of reptilian behavior, or so-called behavioral sedentarism. These beliefs have shaped perceptions of snakes as animals that mattered less, long thought to be incapable of complex thought or emotion, and hence unable to meaningfully suffer. However, these long-standing biases are increasingly being actively challenged as new research demonstrates that snakes have far more complex sensory and cognitive capacities than was previously acknowledged, giving rise to concern about the impacts that captive conditions can have on their welfare. Snakes show most, if not all, of the spatial cognitive processes of mammals and birds, but on the whole, they perceive the world differently and in fact have some mind-blowing capacities and accordingly have different needs than our more traditional pets. Their response to human presence may obscure behavioural indicators of welfare, so we need to be careful and objective when assessing whether provided conditions are acceptable. Snakes are kept in homes as pets, in zoos and wildlife parks, in research facilities, traded around the world both legally and illegally, and as rescued and rehabilitating wild animals. A 2019 quantitative study of Australian pet owners estimated that pet snakes were kept in almost 50,000 households. When asked the main reason for acquiring a pet reptile, the most common response was that they were believed to have lower maintenance requirements than other types of pets. In contrast, it is well documented that the most common health conditions in captive snakes are the result of inadequate husbandry. Exploring captive mortality rates and levels of owner knowledge reveals some troubling statistics. Several authors have estimated the first-year mortality rates of companion reptiles in UK homes, and the results vary wildly from 3.6% when self-reported by owners to up to 75% via other methods. This huge variation suggests a lack of robust data, perhaps in part due to deficient record keeping and a historical lack of scrutiny and a fear of legal repercussions. In a survey of 220 Portuguese pet reptile owners, 85% failed to provide at least one of 4 basic husbandry needs, despite rating their pets' welfare as good or very good. Generally, the owners were capable of recognizing the behaviors associated with poor welfare and captivity stress. However, 25% considered these behaviors normal or natural. Other research shows many owners underestimate the likely lifetime total cost and time commitment of their pet reptile. So what is going wrong here? Well, let's think about snake welfare from the perspective of the 5 domains model. Here we consider the 4 functional domains of nutrition, physical environment, health, and behavioural interactions as influencing and allowing us to infer the 5th domain, the mental, emotional, or affective state, something we are only just starting to appreciate when it comes to reptiles. Reptiles lack the facial expressions and many of the behaviours that accompany emotion and distress in mammals and birds, but this does not mean they lack emotions or do not suffer. Recent research has demonstrated that there are pronounced personality differences between individual snakes of the same species. For many captive animals, carers can satisfactorily address domains 1 to 3, and the focus for enhancing welfare is on domains 4 and 5. For snakes, fundamental aspects of domain 2, their physical environment, are frequently lacking. The deficiency significantly overlaps with other domains, especially domain 3, i.e. their physical health. So let's explore this a little further. As ectothermic poikilotherms, every aspect of the life of a snake is governed by its daily experience of solar light and heat. Snakes derive the vast amount of their body heat from external sources and primarily regulate their body temperature fluctuations within a given range. Body temperature affects most aspects of their physiology, behavior, and health status. They control their body temperature by behavioral means, utilizing thermal gradients within their environment, i.e., they have an innate need to perform behavioral thermoregulation. Research has shown that long-term maintenance of most snakes at so-called room temperature is harmful. Too cold for normal digestion and immune function, too hot for brumation of temperate species. So in terms of environmental needs, the maintenance of a range of environmental temperatures within the preferred optimal temperature zone is critical for snakes to behaviorally thermoregulate. Light, both visible and contrary to our previous understanding, ultraviolet, must be provided for snakes to express circadian behavioral cycles and normal basking behavior. Other critically important environmental elements are photoperiod, humidity, furnishings, substrate, and cage materials. Habitat design for captive snakes is thus an important skill especially as each species has particular requirements. Because of this need to be maintained within a space that has suitable temperature, lighting and humidity, and unlike more common companion animals, most captive snakes live the vast majority of their lives in enclosures. This brings us to one of the most fundamental issues of environment: the dimensions of the space afforded to them.
Can you think of any other animal that we would propose housing for the entirety of its lifespan in a space that did not permit it to stretch out fully? It might seem like a ridiculous suggestion, but this is the accepted practice of housing captive snakes. Snake captive environments typically involve enclosures with dimensions that prevent their occupants from adopting a straight line body posture. In particular, in the commercial, hobby and pet sectors, it is very common practice to keep snakes in small barren boxes or plastic tubs, often stacked in racks, with just a heat lamp, a clean water source, a sloughing aid, and easily cleaned plastic or paper-lined flooring. Many zoos and other facilities maintain at least some of their snakes under similar conditions. Captive snakes may be the only vertebrates where management commonly involves deprivation of the ability and welfare need to freely extend the body to its natural full length. We know that even so-called sedentary species of snakes utilise significant space as part of their normal, natural lifestyles. A recent study aimed to ascertain whether enclosure size impacted on the behaviour and welfare of captive corn snakes. 12 snakes were housed in enclosures that were either 2/3 of their length, designated small, or longer than the length of the snake, designated large, for around 32 days. Their welfare was assessed through observation and behavioural tests. Then each animal was switched to the other housing condition and received the same welfare assessments. When housed in large enclosures, snakes were more active and spent time stretched out, a resting or comfort behaviour not possible in the small enclosures. Finally, all animals received a preference test. When given a choice, snakes showed a clear preference for the large enclosure whilst active, although this preference was not observed whilst resting. These findings suggest that providing captive snakes with larger enclosures is beneficial to their behaviour and welfare. In another recent study of 35 ball pythons, 2 housing conditions were compared: furnished terrariums and a traditional rack system with unfurnished tubs, both with automatic temperature control. In the racks, the pythons had a hiding place and a water bowl, and the lighting in the room served as indirect illumination. In contrast, each furnished terrarium size was correlated with the body length of the python it held and contained substrate, a hiding place, climbing possibilities, a water basin for bathing, an elevated basking spot, and living plants. Illumination was provided by a fluorescent tube and a UV lamp. The behaviour spectrum differed significantly between the 2 housing systems. The pythons were more active in the furnished terrariums. Basking, climbing, burrowing and bathing could only be expressed in the terrariums. Abnormal behaviours that could indicate stereotypies were almost exclusively seen in the racked tubs. The results showed that the housing of ball pythons in unfurnished tubs leads to a considerable restriction in species-typical behaviour. The assumption that snakes are content to remain coiled permanently in the space available is now outdated and not supported by mounting scientific evidence. Not providing sufficient space to allow snakes to fully elongate or adopt a rectilinear posture appears to thwart a behavioural need and thus impacts negatively on their welfare. You can see in the image on the left a python attempting to adopt a rectilinear posture while confined to a small box at a pet fair. Obesity, dystocia due to poor muscle tone, constipation and spinal disease are all common in captive snakes and may well be associated, at least in part, with their extreme confinement. Traditional practices reflect snakes' inconvenient shape rather than what is good for them. Reptile welfare is enhanced as much by environmental enrichment as is the welfare of other groups of animals. Well-designed enrichment programs provide behavioural and physiological benefit, increase activity, lower body weight in those species prone to obesity, and improve reproductive function. The aim of such programs is to promote normal behaviours including foraging for food and water, thermoregulating, and seeking shelter or mates. In the image on the right, you can see a graph demonstrating the preference from baseline that Western hognose snakes show for environmental enrichment, which increases over time and unexpectedly is even more appealing when on the cooler side of the enclosure, shown by the dotted blue line. Although declarations of sentience often include the term all vertebrates, snakes are often accidentally overlooked or ignored when it comes to applying this concept. Snakes possess all of the necessary capacities to be classified as sentient beings, at least in the species that have been empirically investigated. Our knowledge of snake behavioural complexity is rapidly increasing, with at least 24 behavioural signs of captivity stress having been described. Modern zoos and progressive private owners are keeping abreast of the immense knowledge base required to meet the environmental needs of the myriad of snake species kept in captivity, and are developing solutions to this challenge. Increasingly sophisticated snake training programs are being established, and this is giving these animals some long overdue choice and control, and is helping to minimise stress. In the image on the right, you can see a venomous rattlesnake participating in a training session using a target ball, which allows the snake to be moved between enclosures without having to be handled. This is less stressful and safer for both animal and keeper. Some learnings from this program were that training sessions typically take longer than they would with a mammal, that snake behaviour is very subtle, and that snakes perceive scent and temperature significantly more than visual movement. So in summary, traditional practices reflect snakes' inconvenient shape rather than their unique biology. A failure to adequately provide for their range of physical and behavioural needs can be fairly described as controlled deprivation. Science and progressive snake carers and advocates are rightfully questioning the attitudes that these animals are simple, low maintenance, sedentary and easy to care for. Their needs are different, not lesser, and it is time to address our biases and apply new learnings. Innovative keeping developments are demonstrating snakes' unique behavioural and cognitive capacities and how these can be supported via novel methods of enrichment and housing design. Thank you so much for your attention, and we've included the references from the presentation here.
Thank you very much, Michelle, and also to co-presenter Matthew Crane. Very fascinating topic. I'm really looking forward to the Q&A session that we have at the end. Okay, now I'd like to introduce our final speaker for this 7th session, and I'd love to introduce Megan Lucas from the Animal Welfare Science Centre at the University of Melbourne in Australia. And Megan is going to be talking about piglets, piglet early life housing experiences and impacts on stress resilience. Thank you, Megan.
Good morning and good afternoon, everyone. I'd like to start today by acknowledging the traditional owners on the land on which I normally work. That's the Wurundjeri people of the Kulin Nation. I'd like to pay my respects to their elders, past, present and emerging. My name is Megan Lucas and I'm a postdoctoral research fellow at the Animal Welfare Science Centre at the University of Melbourne. And today I'm going to talk about a little bit of research that was conducted as part of an Australian Research Council linkage project on early experiences and stress resilience in pigs. I'll give you a little bit of a spoiler alert on the research that I'll talk about today. It was somewhat controversial in that we actually found some positive benefits in terms of welfare of varying crates for pigs. So I'd like to start off today by talking about stress. Now, stress is something that we all encounter to one extent or another on a daily basis. And for the life of a commercial pig, it's really no different. Pigs encounter a number of routine challenges as part of routine management and production in a commercial system. Now, an impaired ability to cope with these routine challenges has implications for pig welfare and also for pig productivity. And a pig who has a really strong ability to cope with these routine stressors would be referred to as a stress resilient pig. Now, stress resilience is something that we know is shaped by early life experiences when the brain and the nervous system are extremely plastic and amenable to change. In particular, stress resilience is shaped by early life experiences with stress, whereby variations in the amount and intensity of early life stress exposure shifts animals towards becoming vulnerable or resilient to stressors throughout their life. So essentially, moderate or mild stress during the early life period leads to stress resilience by allowing animals to form adaptive coping strategies for future stressful situations. So of course, all of us academics are sitting up here on the top of the peak of the curve. On the other end of the spectrum, severe or prolonged or intense early life stress exposure frequently dysregulates physiological stress coping mechanisms and leads to stress vulnerability or an impaired ability to cope with stress. And similarly, a lack of stress exposure altogether also leads to stress vulnerability because it leaves too few opportunities for animals to learn to cope with stress effectively. Now for piglets, there are a number of early life experiences that are likely to provide differing levels of challenge and therefore are likely to affect the development of stress resilience. So in the experiment that I'm talking about today, we were interested in studying the effects of early interactions with humans and also the effects of the early housing system on stress resilience, since the housing system actually moderates a number of key other early experiences, including the interactions between sows and piglets, between pigs and other pigs, and overall the complexity of the environment and how many opportunities for learning and how many challenges it offers animals. Now, in Australia, something like around 90% of our pigs are reared in indoor systems in farrowing crates. Now, for piglets, there are some benefits of these systems since they are known to reduce the mortality risk from being crushed by the sow. However, there are obvious welfare concerns for the sow since her behaviour is highly restricted. She can stand up and lay down, but she is restricted from turning around. And this has led to the design and research of a number of alternative farrowing and lactation systems that allow more freedom of movement for the sow. Now, while we know that sow welfare is superior in some of these loose systems, there has been less research on the welfare implications for the piglets. And in particular, there has been overall, I suppose, a lack of research on longer term effects. So some studies have looked at short-term welfare benefits of these loose farrowing and lactation systems for piglets, but overall there's a lack of research on lifelong effects or effects after weaning. So in this experiment, we were interested in rearing pigs in one of two farrowing and lactation housing systems, and they were both very similar to the pictures that you've just seen there. Either a standard farrowing crate where the sow was confined throughout the farrowing and lactation period, or a loose farrowing and lactation pen where the sow was free to move, and therefore piglets had greater opportunity for maternal care and interaction with the sow. Piglets also had a much greater space, and they had more physical complexity in their environment just through design features in the pen. And this was a factorial experiment, so within each housing system, piglets were allocated to one of two human contact treatments. They either received routine contact from stock people, which was just human contact associated with regular husbandry and management, or they received positive human contact, which involved routine contact plus a regular opportunity during the lactation period to interact with a person and be scratched, patted and stroked by them. So our handling and housing treatments were imposed from 0 to 4 weeks of age during the lactation period. And after weaning, all pigs were housed in identical pens under the same stocking densities. They had no enrichment and they were managed by the same people. We collected a number of measurements of stress resilience until 21 weeks of age when pigs were sent to market. And these measurements included responses to challenging situations such as close human contact, isolation and novelty, responses to the normal environment to assess general coping ability, so for example basal stress physiology and time budgets of behaviour. And then we also looked at the consequences of coping with stress on animal health and productivity. So I'll talk about some results today. My focus is really going to be on the effects of the early housing system. I did mention that this was a factorial and we looked at the effects of early human interaction as well. But today, just in the interest of time, I'll focus on the housing system. But I did want to point out that there were a couple of interactions between our two effects. So I'll raise those when necessary. So starting off with our time budgets of behaviour. During the lactation period, we found that loose pen piglets were more frequently observed initiating interactions with the sow, performing play behaviour, performing aggressive behaviour as well, although this was likely conducted in conjunction with play behaviour, And they were less likely to be observed repetitive nosing of pen mates. So they were less likely to show behaviour such as belly nosing, which is an abnormal behaviour we see pigs show. However, there were very few effects of the early housing system on time budgets of behaviour after weaning at 8 and 12 weeks of age. Now, we looked at some measures of basal stress physiology, and one of the markers that we looked at was BDNF, brain-derived neurotrophic factor. So BDNF is a neurotrophin, and it plays a really important role in learning and memory, and it's associated with stress resilience. It hasn't been extensively studied in pigs, but there are a handful of studies on BDNF. To give you an example, we know in pigs that it increases with the provision of enrichment. Now, we found considerably higher levels of BDNF in our loose pen pigs during the treatment period or pre-weaning compared to in our farrier crate pigs. Now, in rodents, we know that BDNF is associated with higher levels of maternal care, improved spatial navigation, and greater levels of social play. So it wasn't totally unsurprising to see higher BDNF in our loose pen pigs during the treatment period because of those factors. However, there were very few, or no effects rather, on our BDNF after weaning, with levels being quite similar at 8, 12 and 17 weeks of age across our two housing systems. Now, I will point out here, although it's not very important, you'll notice that BDNF decreases with age. That was an expected finding since we know that BDNF has an important role in brain plasticity, which is very intense early in life and reduces with age. Now, something else about BDNF is that we know that it's thought or hypothesized to mediate stress resilience by regulating HPA axis activity early in life. But despite seeing high BDNF in our loose pen pigs early in life, we didn't actually see any effects on cortisol concentrations at any age. Cortisol concentrations in all groups appeared to be slightly higher at 8 weeks of age, and there was a tendency for higher cortisol in loose pen pigs at that time, but the effect was very small, so it was unlikely to mean anything significant. Now, despite some of those positive effects of our loose housing treatment during the lactation period, so increased BDNF levels, increased play behaviour, and less repetitive nosing of pen mates, we actually saw that our loose pen pigs showed greater fear and greater stress responses to a number of situations. So to give you some examples, during the treatment period at 3 weeks of age, loose pen pigs showed more avoidance and therefore more fear of novelty. So here we used a novel object, which was a traffic cone, and also more fear or more avoidance of humans as well. And that more avoidance was evident by our farrier crate pigs being faster to, or rather less avoidance in our farrier crate pigs was evident by them being faster to approach and faster to interact with the novel stimulus and with the human stimulus. And they also showed greater numbers of interactions and spent more time in close proximity with both of those stimuli as well. Now, there was some evidence of reduced fear of humans in our farrier crate pigs after weaning. So, for example, our farrier crate pigs showed reduced proximity to a moving human in their home pens at 6 weeks of age. However, there were no detected effects on fear responses to humans at 9 weeks of age or at 14 weeks of age, indicating some loss of the effective early housing system on fear responses to humans. At 7 weeks of age, we tested pigs with a period of 2 minutes of social isolation in an arena outside their home pen. We didn't really see any effects on of the housing system on escape attempts from the arena, but as you can see, escape attempts were quite rare. It was less than 1% of farrier crate pigs that attempted escape from the arena. We did see some indication that our farrier crate pigs, or rather our loose pen pigs, were more likely to vocalise and more likely to vocalise sooner compared to our farrier crate pigs as well. And when we look at serum cortisol concentrations 45 minutes after the isolation period, cortisol was lowest in our farrier crate pigs, particularly if they were in the positive human contact treatment as well. Now, additionally, we found that pigs reared in the loose pens showed greater escape behaviour when they were being captured for processing at 4 days of age. They had a greater cortisol response 2 hours after weaning, although not at 49 hours after weaning. And there were a few more interactions with our human contact treatment after weaning as well. So for example, loose pen pigs from the routine human contact treatment showed greater levels of escape behaviour and greater repetitive nosing directed towards pen mates. So for example, belly nosing in that immediate period from 0 to 1 hours after weaning. And additionally, at 21 weeks of age, we found that farrowing crate pigs were, or rather loose pen pigs, were more likely to balk or more likely to hesitate when they were being moved out of the home pen at 21 weeks of age, which was quite significant. Not unsurprisingly, we found greater levels of pre-weaning mortality in the loose system, most likely due to increased crushing, but we didn't record, unfortunately, specific causes of mortality. The greater pre-weaning mortality in the loose system led to smaller litter sizes in the loose system, which was probably one contributing factor to the loose pen piglets having higher growth rates during the lactation period. We didn't see any effect on of the housing treatment on growth after weaning when all pigs had equal stocking densities and management. And we also didn't see any effect of the early housing system on pigs' injuries at 4, 5 or 17 weeks of age. So to summarise some of these key findings, the early housing environment in this research had quite a considerable effect on the fear responses of pigs, particularly early in life, and on the human-animal relationship. However, there are very few effects of the early housing system on pig behaviour in terms of time budgets of behaviour, basal physiology, and on fitness after weaning. Now, the loose pen pigs showed greater levels of play behaviour. They had they initiated more interactions with the sow. They performed less repetitive nosing of pen mates, and they had higher BDNF concentrations during the lactation period. And interpreted together, that evidence is really showing us that the loose pen pigs had superior welfare in their undisturbed home pens during the lactation period. However, they were clearly less resilient to additional challenges in their environment based off increased stress responses and fear responses to humans, novelty, isolation, weaning, and routine husbandry procedures. Now, one explanation for the increased stress response of loose pen pigs is that they were reared in a more isolated environment due to the loose pen design and shed layout. So to give you an example, the high walls of the loose pens actually restricted piglets' visual contact outside of the pen, whereas farrowing crate pigs could see people walking past quite regularly. And there were several other reasons that the environment was more isolated in the loose pen system as well, which I'm happy to expand on. And so this more isolated environment and this reduced exposure to stock people, other pigs, and less visual stimulation in general in the loose system may have actually led our loose pen pigs with too few opportunities to learn to cope with challenge. So perhaps they're sitting on this end of our resilience and vulnerability spectrum. It is also possible that the maternal behaviour of sows in the loose system played a role. We know that in the loose system, maternal responsiveness is increased, and heightened protective maternal behaviours by sows when stock people are close could actually increase pigs' fear of stock people. Thanks very much. I'd like to acknowledge all of my wonderful co-authors that are up on that last slide there, and of course our industry partners.
Perfect, thank you very much. And we are now going to move to our Q&A session. So if I could have all of our panelists join me. Perfect. Okay, now, sorry, let me just shuffle my view. So my first question is going to be for Gloeta. And this is a question that's come from A. Rutherford. Can everyone hear me okay? Yep, perfect. My first question has come from A. Rutherford, and it's for Gloeta. And the question is, Gloeta, were you able to explore the reasons for euthanasia and correlation to wait times? For example, lorikeets with highly infectious beak and feather disease that is not necessarily painful, but not usually releasable.
That's a really great question. So the wait times, basically, to put it in perspective, for them to know that that bird had beak and feather disease, they had to take the bird out and handle it. And those wait times also correlate with how long it generally took before they were even taken out of a box and inspected. So at 31 minutes, that's when they discover that the bird has beak and feather, right? So the initial handling, almost all of the animals are being handled under anesthesia, because to do any sort of triage, I mean, there is some triage, don't get me wrong, but to do any sort of intensive triage requires the animal to be under anesthesia. We do see a species difference. So for sure, some species are taking significantly longer to be treated than other species. And so we definitely see a bias within the data towards particular species. I'm not going to name them because that'll come out later, but I can say that if you happen to want to come back in your next life, don't come back as a rainbow lorikeet. Not if you want to go through a speedy process.
Thank you. Okay, so my next question will be for Michelle and Matthew. This is going to come from the chat as well, also from A. Rutherford. And their question was, do you have any insights into why captive snakes often burn themselves on heat lamps rather than moving off them before being burned? Actually, I think, oh, yeah.
I'm unmuted. Thank you for the question. Yeah, it's a great question, and I think it is a really good example of situations where, if we try and extrapolate from what we understand about the welfare and the health needs of animals that we're a bit more familiar with, like mammals, that we can make some really we can make some assumptions that can end up being a bit of a problem. So we know that we do know that burns from inappropriate heating sources do occur quite regularly in captive care, and the likelihood is that the neuroreceptors in the skin of many of the reptiles that are kept in care are very different from the receptors that we have on our own skin. And as you can imagine, in the wild, it's very unlikely for a reptile to end up in a position where they're in contact with something that is exceedingly hot, hotter than would occur in nature. And if so, they would have the freedom to move away from that. So whilst they have heat-seeking behaviours, the detection of a dangerous level of heat is likely to be impaired just through their normal biology. So really, a really significant and serious problem, and one that can certainly be prevented by good husbandry, good heating sources with adequate protection around them. So yeah, it's certainly, as I say, a good example of that assumption that's been made about what we know about other animals that would choose to move away.
Yeah, sure. My next question is for Megan. Megan, this comes from Mia Cobb. And the question is, could the increased escape behaviour that you've seen be a functional or adaptive advantage that's been learned from having access to more space or more spatial awareness? So could it be that rather than being more resilient, the created piglets are more in learned helplessness mode without the benefits of agency within the space?
That could be one possible interpretation. I suppose we've got a lot of measurements that we're interpreting all together here. I think we heard in Sarah's talk this morning how when we're assessing welfare and stress resilience, we can't just rely on one single measure. And here we're, I suppose, trying to paint a picture of what's going on by looking at not just escape behaviour, but also their vocalizations, their cortisol concentrations, and their actual physical reactions to being held as well. So when we're interpreting all of those responses together, it's indicating a greater stress response to those challenges, which we interpret as reduced resilience. I'm not sure if I fully answered your question there, Mia.
Okay, we are going to go back to Gloeta. Let me just find the question I was going to ask you though. Okay, so this question comes from Cullen Brown, and he has asked, In many instances, the best outcome could be immediate euthanasia, but your average person is unlikely to be capable. What do you think is a solution from your research?
Yeah, that's a terrific question. There were times during my research where I thought the best thing I could do was stop my research and develop a way to euthanize animals in the field like this that your average person could use. The simple fact of the matter is that we don't have anything like that right now. Either we have drugs that make the animal toxic to anything that eats it, or you're asking people to carry around sledgehammers and guns, which isn't something that your average person in the Gold Coast is going to do to a lorikeet. And so how do we ensure that a rescued animal, if it can't be euthanized immediately, once it arrives into the system, that it receives the most rapid care possible? And the problem is the system as is completely overwhelmed by the number of animals coming in. I mean, we're seeing an increase every year. So between 2002 to now, we went from basically 5,000 animals a year being rescued in Southeast Queensland to these hospitals to over 50,000 being rescued. And that number is increasing every year. And the time that the animals are coming in. So maybe before you would see 40 animals coming in for 4 vets, and now you're seeing 120 animals coming in for 4 vets, because it's not a federally supported, it's not a state supported system. So it's all privately or gift supported. And so you have a whole system. I mean, the comparison would be to the human hospital system. What happens when you don't fund it? What happens when you don't have enough staff? What happens when you overwhelm the system? What does that do? So I guess in that sense, if we can't euthanize them in the field, how do we make sure that the staff are supported? I mean, there's that little side thought of mine is maybe people would pay more attention to the environment if they actually had to see all the bodies on the side of the road from road strikes, right? So maybe we could actually start to have those conversations.
Okay, I'm going to, Michelle, did you have a question?
I was just going to add something to that, if I may.
Absolutely, sure,
fantastic research, Gloeta, really overdue. But just with my veterinary hat on, I guess another consideration is that for quite a number of these animals, and I'd be interested if this was your impression from your observations, Gloeta, that for many of these animals, they require quite a detailed examination and potentially some diagnostic testing, like, for example, an X-ray, to be able to determine whether euthanasia is the appropriate course of action. And of course, delivering that in a field situation can be really difficult. So was that the case with some of yours, the cases that you witnessed?
Yeah, that's a really great question, Michelle. So the big thing is that I would say that most of the animals get X-rayed, like just as part for the course, which is part of the system that slows down. But the problem is, at least I don't know for New South Wales, in Queensland, the law is very clear that if the system is burdened, euthanasia is the default. Like our code of ethics for Queensland wildlife rescue says that the aim of wildlife rescue is to stop suffering. It's not to save lives. And so that's a really distinctive difference in how people picture wildlife rescue. So you have animals coming in, let's say a rainbow lorikeet with, rainbow lorikeet paralysis syndrome. Like that bird's probably not going to get X-rayed. The bird, depending on how paralyzed it is going to be euthanized, like simply because there's no room for that bird. And so it won't ever need, like they could have compound fractures, but you wouldn't know because the decision to euthanize is already in place just from that initial investigation. So I don't know if that makes sense as an answer, Michelle, but like stepping back and it's not even because of X-rays that most of the animals are getting euthanized. Most of the animals are getting euthanized because there's no way, there's not enough carers, there's not enough support, there's not enough facilities, there's not enough staff, there's not enough training. So it just becomes the system.
Hi guys, we're at that time where the session has flown. So I want to thank all of our speakers and Chris for being a wonderful chair for today.
My pleasure.
Kat and I have really enjoyed the conversation, and again, the diversity of talks that we get at UFAW events is always just so interesting. You never know where you're going to learn something that you can apply in your own work or hear of a new measure or strategy that you can apply. So just really appreciate all of you coming and sharing the work that you've been doing today. We hope that you'll join us tomorrow. We've got some exciting sessions. We're looking at things like how can we afford animals' moral standing, looking at change management in practice, and also some topics or talks relating to companion animal welfare. I'll throw to Kat to sign off for the day.
Thanks, Mia. Yeah, I can't add much more than that, other than it's been a really great day, and thanks to everyone that's attended. Thanks to our panelists and our chairs for doing such a great job. Couldn't be happier with how it's gone. Thank you so much to everyone. I'm just going to sign off by saying mati wā, or see you later. See you soon. Bye.
Bye.
Thanks, everyone. Bye.